So I'm going to have some hierarchical Bayesian modeling in here, and specifically it's going to be in the context of this stuff called Approximate Bayesian computation, which many of you have probably heard about. But the big picture, just to get into some more sort of big picture things here, I'm interested in reconstructing communities and assemblages in time and space. And I use the word community really loosely. It could be something like the avian assemblage in the neotropics, you know, or it could be something like, you know, a network of associated species, such as like oak trees, parasitoid gall wasps, and the parasitoids of the parasitoid gall wasps. So, and my general approach here is to use aggregate population genetic data to kind of make inferences about multi-species demographic histories. So, and that can be anything from collecting DNA barcodes from large numbers of non-model species, or it can be, you know, collecting smaller numbers of whole genomes or partial genomes from taxa that are often co-distributed and have a similar ecological context. So, and then, you know, I generally use a model-based approach. You know, a statistical model that can generate distributions of data. In this case, again, comparative population genetic data, so aggregated population genetic data sets that we pool for multiple species, and then, you know, eventually we kind of got to move backwards to infer something about these historical models. And I should just say that this has kind of been a pipe dream from the early days of phylogeography and comparative phylogeography to make aggregate species demographic histories. One of the original ideas is that, you know, sure, we can collect mitochondrial data from large numbers of species. We can make historical inferences from each of these species, and if we kind of pull all this together, then we can, you know, answer questions that are interesting to community ecologists in macro, you know, in the context of macroecology. You know, how have whole communities responded to past periods of climate change and changes in the landscape? You know, and just a couple of, you know, a list of possible questions you could get at. Is vicariance or dispersal important factors in the diversification of whole assemblages? What are the relative contributions of stochastic and deterministic factors for community assembly or disassembly? And then thirdly, and I'm going to actually kind of emphasize this for the remainder of the talk, how historically cohesive do communities expand or contract over time? So I kind of see this as coming in sort of a couple of different flavors, at least now in the literature. Now we're able to collect whole genomes or partial genomes from, you know, pretty much any non-model taxon. You know, and you'll often see this in the literature, inferences of population size over time using genome data, and often researchers will overlay these estimated population size trajectories and make sort of, you know, inferences about how these different species may have responded to past climate events in the last, let's say, 50 to 100,000, a million years or so. Another flavor that you'll see, and I think that this group is particularly interested in this type of integrative approach, so bringing in a number of different data types. This is a study that came out a few years ago with the general question of about megafauna and what caused their extinctions, either locally or globally. Was it climate change or was it, you know, human hunting? And so this type of study, you know, may use ancient DNA, paleontological data, human archaeological data, but also importantly genetic data, and also using a model-based approach to make inferences about population size trajectories locally and globally. However, I've been sort of focusing on another approach. I tend to call it the sort of the mega mitochondrial approach, but also the ingredient that I bring in here is hierarchical modeling. And I'm going to talk about a study that we recently worked on about the community expansion and assembly of the Eastern Nearctic. So this recently came out. This is a study involving a number of New York City-based people Frank Burbrink and Brian Smith, also Yvonne Chan from University of Hawaii. The type of data set we were dealing with here was from a number of different vertebrate lineages, about 90 in total, and we're just dealing with mitochondrial data, let's say 10 to 500 individuals per lineage, and we're going to use a hierarchical Bayesian model that I'll explain in a second. So again, what a lot of people have done with these kinds of data is basically use for every particular species, make an inference about population size change over time, and then just sort of overlay everything. And this is kind of what you would get, at least in this case with the Eastern Nearctic data. It's kind of a big mess. It's a little bit impractical for large numbers of species. It's difficult to make to statistically compare results. We want to kind of put this in a sort of a global model to make inferences about the entire assemblage. At least we want to try to do that. And so we turned to a hierarchical Bayesian model, and these are good. They're flexible. We can incorporate within species uncertainty as much as we want, but we can also make across species estimates. And again, I want to emphasize the flexibility here. I'm just going to sort of go through one particular hierarchical model that we've done, but I want to just, it can be done in a lot of different ways. We can possibly construct these hierarchical Bayesian models using estimates from completely different data sources like paleopollen. That would be pretty exciting, I think. So in this particular case, we're focusing on the timing of expansion. So how did all these lineages expand into the northern area of eastern North America? How do they do that over time? And how cohesive was this? So we were sort of keeping track of this hyperparameter. Basically, it's sort of just the amount of the community that co-expanded at a single time in this simple case here. Also, we might want to estimate the timing of co-expansion. So we have all these other sort of lower level parameters that are sort of fed their prior distributions from these hyperpriors. So we have all the different expansion times. We also have all these sort of more nuisance parameters we may not be focusing on, but we could focus on them if we wanted to. But the expansion magnitudes, the effective population sizes, how these effective population sizes changed over time, those are all free to vary across species. But we're also able to estimate the amount of variability across species in these particular lower level parameters. It's just kind of like a more cartoon depiction of how this works. We're modeling how the population change over time, and we're also modeling how the genetic data look given all these different histories. So here we have these, a group of populations that expand over time, but in this case they're expanding at the same time. In this case they're all expanding at different times. Here's sort of a mixed situation. Again, this is just sort of a simple situation here. We could have multiple co-expansion times if we wanted to. We could have contraction times. We could have extended bottlenecks. But we're doing this, and we're doing this across 90 or so lineages. And importantly, in any particular species, we have to make sure that the unit that we're feeding in is really a panmictic population, a free mixing population. So if we take a species of, let's say, snakes, it may consist of, like, say, 2 or 3 freely mixing but rather isolated populations. We would split that into 3 different lineages. If we look at a species of birds, it may just be one big, freely mixing population. That would just be one unit in this case. So, this is our hierarchical Bayesian model. It's a little bit difficult to deal with analytically, and of course we want flexibility. We want to try a bunch of different models. So we turn to this tool, Approximate Bayesian computation. And the general idea here is that the likelihood of a parameter or hyperparameter, the value is inversely proportional to the difference between data that you simulate and data that you actually observe. Of course, we're dealing with genetic data collected from many different individuals across a whole bunch of different species. We have good simulation models. We're using the coalescent in this case. It's a standard simulation tool for population genetics, but it's very difficult to really hit the data exactly. So again, this approximate Bayesian computation approach really comes in handy. And this is just sort of a flow chart of how it works. If we simulate the data, and then if we rely on something, sort of a data compression method, so just summary statistics, that makes it much more manageable to actually hit the data with our simulations. And the idea here is that the data that we simulate that is very close to the data that we observe must come from parameters that generated the data. So again, you have this sort of flow chart. You do this iteratively many, many times, and you really keep track of the data that's very close, the simulated data is very close to the observed data. Possibly, and again, generally, we're actually trying to solve this sort of Bayesian Bayes theorem here. And I think this is like sort of one of my more favorite schematics of how ABC works. This is from Mark Beaumont. You can think of a simulation model where you're drawing parameter values across your prior distributions, and then you're generating data according to that model, and you come up with a big sort of pile of data here. You have these contours here. And you kind of take a snow plow and you sort of plow all the snow here onto this side. Here you get your prior distribution. Here you could plow it over into this direction. You would get this sort of probability of data. And you could take a slice here. This is your likelihood slice, probability of the data, given the parameter. And then, but this is where what we really want to do. We have observed data. Let's say it falls in right here. This slice right here is sort of our posterior distribution. And of course, it's really hard to hit the data exactly, but you can measure how close you are to the data very easily using summary statistics. So we use some kind of like threshold here, and we come up with this approximate posterior distribution. So back to the Eastern Nearctic, we do this across all the different lineages. We generate, we basically, at the very end, what did we get in this case? Well, it looks as if the expansion was incredibly asynchronous in general. However, a couple of nuggets did fall out. We do find that all of the snakes basically co-expanded very recently, only in the last 15,000 years or so. Lineages, most of the frogs and mammals, however, cohesively co-expanded maybe more about like 120,000 years ago. The other lineages were really all over the map. That's sort of the take-home from this particular study. Now, a couple of very important things that are missing here. First of all, we're just using one locus per species. There's a lot of stochasticity associated just with one mitochondrial locus. We now, a PhD student in our lab, Alexander Jouet, he's developed a way to use SNP data across many different species, and also he's expanded this general framework into a software package that can allow for many different kinds of hierarchical multi-species models, not just this co-expansion one that we're talking about here. Another key ingredient here, I can just plug what Diego has been working on. Diego was with us for a couple of years ago, and you can talk to him about this, but a very exciting method referred to as the spatial site frequency spectrum. And here is maybe a good way to explore the spatial origins of community expansion. And I'll just leave it at that. Another area that we are getting into right now is a way to sort of bridge ecological neutral theory with neutral population genetics. And again, I'll be very brief here, but here this is the work from another PhD student in the lab, Isaac Overcast. And here this could be sort of a sort of geographical community assembly in this type of context here of North America, but also like an island chain. A volcanic island series. It could be in any really sort of meta-community situation where you have history and you have some spatial component. And the idea here is to sort of ground comparative phylogeography in sort of classical ecological theory, something that hasn't really been done, to my knowledge, very well. And this could maybe allow us to account for the relative importance of deterministic and stochastic processes governing assemblies. And also, maybe answer questions of, basically a way to sort of jointly produce species abundance distributions, genomic diversity and divergence spectra, so distributions of genetic diversity and divergence, but also across taxa distributions of historical demographic parameters, such as expansion times, contraction times, colonization times, and such. And also maybe to sort of estimate some of these while observing other things. So this sounds like it's kind of a mouthful here, but so far we've been able to, so Isaac has explored this. He has a software package called Gimme SAD. The SAD, that's his strange sense of humor, species abundance distribution. But here we have sort of a forward time model. Excuse me. Well, anyway, it's not in the PDF. We have a forward time model of assembly that can generate a relative species abundance distributions, something that we might have a hard time observing in the field, especially with animal species. And then we reparameterize these into something that makes sense within a coalescent population genetic model. And then we can co-generate diversity spectra across, an assemblage of taxa. So that really ends right there. Thank you again, and I'm really excited about talking to everybody and learning a whole lot in this workshop.
Thank you for your talk. This is on, yeah, okay. When doing approximate Bayesian computation, I thought that was like the reason people do that is that the normalizing constant is complicated to compute, and that's the intractability. So I was wondering what likelihood you have in your model that results in being unable to compute the normalizing constant.
Yeah, again, this would be sort of a multi-level likelihood. So we're using, it's calculating the likelihood of a hyperparameter that would be difficult to calculate.
Well,
I mean, that's one general reason, the intractability of calculating the likelihood.
Well, why aren't you just using like some Metropolis Hastings algorithm or Hamiltonian instead?
Yeah, I would love to explore that. Maybe we can discuss that. Yeah.
Okay, thank you.
I'd like to talk more about that.
Sounds like a good group topic or discussion over lunch.
Yep.
The results for the animals here expanding at different times is kind of consistent with what's often seen with plants. Of different plant communities seem to sort of reshuffle as they spread after at least based on some of the pollen data and whatnot. Has anyone looked at like plants and animals? Because it seems like some of those might be where you might expect the pollinator and the plant to go together, or like, something where you have like the snake may not necessarily be interacting with the bird, so there's no reason why they should be expanding together, but that where we know that there are likely to be stronger species mutualisms or predator-prey interactions, things like that.
Oh yeah, we would. Well, yeah. Again, we would love to do plants. We would love to do fungus. The ectomycorrhizal species and everything like that. A lot of this, the fact that we focusing on vertebrates here is a little bit of the legacy of just how much mitochondrial data is available. But I see this kind of more as a first pass. We would like to collect, SNP data from like a much more diverse assemblage, including, tree species. And we have no invertebrates here. But yeah, I agree completely.
So I read the ABC article in Wikipedia the other night, so I know all about it now. Could you say a little bit more about how you choose your summary statistics and the value of epsilon?
Yeah. Well, the summary statistics, these are, in this context of co-expansion, these very well correlate very well with parameters related to expansion. So changes in theta and effective population size. So we're using aspects of average pairwise distances between sampled lineages, aspects of T-Gemas D, different things that correlate very well with population size change. However, we're combining everything across a whole bunch of different species. Those statistics are normally collected at a per population level, so we're also taking the first couple of moments across our entire data set. So we're looking at the variability in those statistics also. I should say, like sort of best practice of ABC is we always go back and validate how well we can estimate things, using basically simulation experiments, and also how well the model can generate the observed data. There's different posterior predictive things. So we're doing all of that good stuff too. And the epsilon, that's another thing you can explore with simulations, to see if you get a good sweet spot. Yep.
So looking at this, I noticed that we're thinking about things in terms of species interactions, which I like them. They're cool. But looking at that plot, I wonder if it's better to think about constraints on the data. So like, if you think about snakes, they're cold. Sorry, they're kind of slow. They don't like it when it's cold. The peak that you have is, I think, sort of around the time of the last glacial maximum. So that it would actually be very hard for snakes to move at different rates just because of the fact that it wasn't very pleasant for them before the time that they all started moving at the same time. So I guess my question is, I wonder if we should be thinking more about constraints, like sort of mathematical constraints on there's sort of the graph starts at 0, it doesn't go past 0, and some things get bunched up, rather than thinking about species interactions or sort of some fancier part of species ecology.
So, yeah, that's an interesting idea. We could constrain things by the prior, I guess, if that's what you're suggesting.
What I mean by constraint is that whatever the probability distribution is for snakes, it has to stop at 0, and it has to have a mean that's close to 0. So basically, you can, it's either it's going to have a peak or it's going to have a long tail, but there are a bunch of shapes that you can't get just because of the fact that the average is close to 0 for that group.
Okay, yeah.
So I think Will is saying that the range of expansion times that are even possible for snakes is different than the range of expansion times that are possible for birds.
Yeah.
Right?
Yeah, I feel comfortable to kind of like let the data speak, and some, priors that are a little bit too restrictive. At some point it kind of turns into a model that you're testing rather than a prior that you're using to estimate parameters. But, we could go that route also. There's a lot of, it's very flexible. So.